Hello, on behalf of the Biochemical Society and Portland Press, I'm pleased to welcome you to today's webinar, which is part of our dedicated Biochemistry Focus ECR webinar series. Topics in this series include different research areas in the molecular biosciences, as well as practical sessions to support career development. Each webinar will give you the opportunity to ask questions via text, and we welcome suggestions for future topics and speakers to feature in our webinar series. So please see the website for more details. My name is Dominika Gruszka. I am the Early Career Member Representative Trustee and Chair of the Early Career Advisory Panel of the Biochemical Society. I am also a postdoctoral training fellow at the Francis Crick Institute in London, where I use single molecule techniques to study the molecular mechanisms of DNA replication and chromatin maintenance. Today's webinar is titled Biochemistry One Molecule at a Time and is part of our dedicated early career research program of webinars. So before introducing the topic of the session, I would like to thank PicoQuant for sponsoring this webinar. PicoQuant is a company which has been growing organically together with the single molecule community for the last 25 years. Their microtime to 100 confocal microscopy platform is considered to be the most open, commercially available system with trusted quality for time-resolved single molecule, FLIM, and FCS applications. Please see their website for more information. And now, before introducing our invited speakers, I would like to play a short introductory video from PicoQuant. So as previously mentioned, today's webinar is titled Biochemistry One Molecule at a Time, and it's linked to a special issue with the same title published in Essays in Biochemistry earlier this year. Conventional biochemical approaches for studying the action of molecules operate on a population level, averaging out any inhomogeneities within the ensemble. By contrast, investigating one biological macromolecule at a time allows researchers to directly probe individual behaviors and thus characterize the intrinsic molecular heterogeneity of the system. Consequently, single molecule methods have unraveled unexpected modes of actions for many seemingly well-characterized biomolecules and often proved instrumental in understanding the intricate mechanistic basis of biological processes. This issue of Essays in Biochemistry showcases different types of single molecule techniques and their applications to investigate processes that are fundamental to life. This is also the first issue written and then edited entirely by early career researchers who helped drive this exciting field of research forward. Today, we will welcome 3 authors from this issue who will provide a sampling of current experimentation using single molecule techniques and the applications of these methods to a range of biochemical problems. So the 3 speakers today are George Cameron from the Francis Crick Institute in London, Dr. Sonja Schmidt from Wageningen University in the Netherlands, and Dr. Peter Heidersen from the University of Iceland. Coinciding with this webinar, we have made their articles freely available to read from 8th until the 12th of November, so please visit the website if you're interested in reading their full articles, and I strongly encourage you to do so. And in fact, George's article is already open access, so you can read it whenever you wish. So before we start, just a short housekeeping note. Questions will be asked after all the speakers' presentations, but you can send in your questions during the talks. So if you have a question, please type it in the question box as shown in the image on the screen, and we will try to answer as many of the questions as possible at the end of the webinar. So without further ado, I would like to now introduce the first speaker of the session, George Cameron. George obtained his bachelor degree in natural sciences from the University of Cambridge, followed by a master's degree in biochemistry, also from Cambridge. Currently, George is the final year PhD student in Hassan Yardimchi's lab at the Francis Crick Institute, where he is studying mechanisms of eukaryotic DNA replication with single molecule techniques. Today, George will show us what happens when the eukaryotic replisome collides with protein barriers. So, all good, George, so I think you can start.
Thanks, Dominika. So I'd first like to introduce to you the mechanism of eukaryotic DNA replication. So billions of base pairs of DNA have to be replicated in eukaryotes. And to replicate this large amount of DNA, replication initiates from multiple origins of replication. The enzyme at the core of the replication reaction is this MCM27 protein and complex. And it is loaded onto double-stranded DNA as an inactive double hexamer in a process known as origin licensing. Later in the cell cycle, CDC45 and GINS proteins are recruited to MCM27, and this leads to origin firing. Double hexamers are remodeled around single-stranded DNA into the CMG complex at this point, and CMG unwinds DNA. Other replosome components are assembled around CMG. During replication, some double hexamers fire whilst others are not used, and the consequence of CMG enzymes encountering double hexamers during replication is not really very clear. But because these collisions between CMG and double hexamers occur somewhat randomly in space and time, single molecule fluorescence microscopy is a useful tool for determining the consequences of these collisions. We can think about possible consequences of CMG colliding with double hexamers. So in reconstituted yeast replication reactions, there's evidence that double hexamers slide ahead of CMG, and it's possible that these provide new origins for firing if the CMG at a replication fork encounters problems. Alternatively, in cells, additional factors might be present, which result in the removal of double hexamers ahead of CMG. And this could ensure unhindered progression of the replosome and CMG through chromatin during replication. So to replicate DNA in vitro in our lab, we use a system of 2 cell-free extracts from xenophus eggs. And these extracts contain all of the necessary machinery for a single synchronized round of DNA replication. We can add DNA to the first extract, HSS, to licensed DNAs, and this occurs in a sequence-independent manner, so double hexamers can be loaded on a range of DNA templates. Adding gemenin to HSS inhibits origin licensing. Adding a second extract, MPE, to licensed DNAs gives origin firing and replication. In NPE, no further origin licensing occurs, and we can also add this protein P27 to stop new origin firing. So we've got a good control of the amount of origin licensing and firing with these 2 extracts. Previous work has used egg extracts to replicate a 50,000 base pair lambda DNA that's tethered to the surface of a glass slide at both ends. And this is visualized with total internal reflection fluorescence microscopy. The DNA is contained inside a microfluidic flow channel to which extracts can be infused and buffers as well. And here's a single labeled lambda DNA with DNA stain visualized by turf microscopy. After tethering the DNAs, we can add the extract to licensed DNAs with double hexamers, and then a replication mix to give origin firing. We'd also add a second replication mix containing the inhibitor P27 kip to prevent further origin firing. And in these replication mixes, we add a labeled version of the lagging strand processing enzyme FEN1. So as replication progresses, FEN1 binds to the nascent DNA, as shown in this movie. And if you're not familiar with a chymogram, below the movie is a chymogram, which is a stack of these images in time with the first time point at the top. And you can clearly see here the growth of a bubble of Fen1 binding to the nascent DNA. In this existing system, however, because the double hexamers and CMG are not directly labeled, we can't look at the consequence of CMG collisions with double hexamers. So I've adapted this system to label MCM3 with a fluorophore. To do this, I immunodepleted endogenous MCM complexes and replaced them with complexes containing recombinant fluorescently labeled MCM3. So after licensing with the fluorescently labeled MCM3, I did a high salt wash to remove any improperly loaded MCMs from the DNA. And you can see along DNAs in a field of view, fluorescent MCMs coating these DNAs. And if we do the same reaction in the presence of geminin, you don't see the same loading of the MCMs on the DNA, suggesting that we're seeing specific loading of the fluorescent protein by the licensing machinery. We can also look at fluorescence intensity traces for the fluorescently labeled MCMs on DNA. And in many cases, we see two-step photobleaching, like here, and in the top right-hand corner as well. We can see proper double hexamer formation has occurred from the photobleaching, two-step photobleaching. To the licensed DNAs, a replication mixture containing labeled Fen1 can be added to trigger origin firing. And we take images every minute to make movies. And a chymogram is also shown below. So here's a nice example where we see a single origin firing, and this double hexamer splitting into two CMG molecules which move in opposite directions whilst unwinding DNA. And the labeled Fen1 binds the nascent DNA between. Replication forks with labeled CMG move at around 400 base pairs per minute on average, which is consistent with previously published work, and this suggests that the labeling of MCM3 doesn't interfere with the function of CMG in the replosome. One possibility I wanted to exclude from these assays was that actually an unlabeled CMG was pushing labeled double hexamers at the replication forks. And to do this, we can use known details of the replication termination reaction. So normally when CMG molecules converge during replication termination, they are removed in a pathway involving the P97 enzyme. But if we perform the same reactions in the presence of P97 inhibitor, CMGs remain on the DNA after convergence and termination. If we imagine the same reactions occurring but with an unlabeled CMG at a replication fork, after convergence and termination in the presence of inhibitor, only one CMG will be seen to be labeled on the DNA after termination. So I performed single molecule replication reactions, in this case with more origin firing and P97 inhibitor, to look at what happens at termination. You can see here that two CMG enzymes approach one another and remain on DNA after termination, suggesting that there is an unlabeled DNA at replication forks. So I next started to look at collisions between CMG and double hexamers. I observed three different primary events occurring. In this first example, we see double hexamer removal occurring. So when CMGs in both directions collide with a double hexamer, you see loss of double hexamer from the DNA. A second type of event we see is double hexamer stalling. So again, labeled CMGs moving in both directions here collide with double hexamers and remain in the same position. So we call this event fork stalling. And the third event we see is double hexamer sliding ahead of the replication fork. So in this example, a labeled replication fork on the right, sorry, an unlabeled replication fork moving on the right-hand side collides with a labeled double hexamer, and then after this collision, the fluorophore continues to move the replication fork, suggesting that the double hexamer is being pushed ahead of the replication. If I count many of these events, I see that double hexamer removal ahead of replication forks is the dominant primary event occurring. It is possible that events interpreted as removal are in fact new origin firing events ahead of CMG, and this is the reason we see so much removal. Another control experiment that was important to do was imaging MCM complexes that have been labelled with a second colour, and this would allow any new origin firing occurring to be observed, as now instead of a magenta CMG moving at the rightward moving fork, it would be replaced with a green labelled CMG. So I licensed DNA with two colours of double hexamers and observed replication. In this example on the left-hand side, you see magenta CMGs in both directions colliding with a green double hexamer and giving removal of these. Whereas on the right-hand side, a green CMG collides with a magenta double hexamer, and this is removed. So together, these results suggest new origin firing is occurring ahead of CMG, and that removal is the most common state of double hexamers when collided by CMG. In reconstituted yeast replication reactions, double hexamers slide ahead of CMG, so an additional factor must be present in extract that results in an assist double hexamer removal. Some recent evidence has suggested that an accessory helicase, which acts on the opposite side of the fork to CMG, might aid double hexamer removal. In egg extract, I'm particularly interested in these two helicases, RTAL1 and FANCJ. To investigate a role for these helicases, they can be immunodepleted from the replication mix. So this Western blot just shows that with antibodies, I'm able to deplete more than 95% of both the RTAL1 and the FANCJ enzymes from the same extract. If we look at the fate now of double hexamers when either RTAL1 or FANCJ are removed individually from extracts, see a mild reduction in the proportion of double hexamers removed ahead of replication forks. However, when we deplete these enzymes together, double hexamers are removed far less efficiently ahead of CMG. I'd just like to say, though, that this data is still fairly preliminary, as it's important to show that what we're seeing is a specific effect of depleting these enzymes. So I'm now in the process of using recombinant RTAL1 and FANCJ to test if defects in double hexamer removal can be rescued. So I hope in this brief presentation I've been able to show you how I've developed a system to visualize single MCM27 molecules during replication in real time. And I've used this system to visualize CMG collisions with double hexamers and shown that double hexamers are mostly removed. I finally showed some data showing that perhaps two accessory helicases are involved in removing double hexamers ahead of CMG and egg extracts. So I'd like to thank my supervisor and other colleagues in the IDIMCI lab for help with the project, and also the CRIC science technology platforms for many parts of the project. So thanks very much for listening.
Excellent. Thank you very much, George, for a wonderful presentation. And now I would like to introduce our second speaker today, Dr. Sonja Schmidt, an assistant professor at Wageningen University in the Netherlands. Sonja studied nanosciences at Basel University and earned her PhD in 2017 from TU Munich, where she developed single molecule fluorescence FRET experiments, invented SMACs, a direct pattern recognition approach to infer single molecule kinetics, and uncovered diverse regulatory effects of protein conformational dynamics. Sonja was awarded an SNF postdoc mobility fellowship and joined Kees Dekker's lab at TU Delft, where she developed a new nanopore-based approach to detect protein dynamics called the NEO-trap. Sonia initiated and now leads the KIN-SOFT Challenge, an international multi-laboratory benchmark study on single-molecule kinetics software. Since 2020, she's been an elected member of the advisory board of the International Threat Community. And in 2021, excitingly, she set up her nanodynamics lab at Wageningen University and Research to develop new optical and electrical single molecule techniques to study biomolecular dynamics beyond current detection limit. And now Sonia will tell us about nanopores, and I believe she will answer the question that she posed in her article in Essays, which states, and I quote, "So why, as a protein fan, should you care about nanopores?" So thank you very much, Sonia. And yes, I can see your slide, so I think we're good to go.
For this kind introduction. Now I think I'm unmuted. Yeah, thank you very much. Also for giving me the opportunity to present our work here. Indeed, my presentation is entitled Nanopores, a versatile tool to study protein dynamics. And as you can guess already from the title and from this slide, indeed, I'm quite interested myself in studying proteins and protein dynamics in particular. And yeah, as you already heard a bit in the introduction, we do this in my newly minted nanodynamics lab, mainly with two favorite techniques. One is single molecule FRET and fluorescence, and the other one is nanopore detection. And in this talk, indeed, I will focus on the second part on nanopores. So what are nanopores? At the basis, nanopores are very simple detectors. They consist of an insulating membrane with a tiny hole inside, as the name implies. This insulating membrane with the hole is immersed in a buffer solution, and if you apply a bias across that membrane, you can measure an ionic current. Now, whenever an analyte molecule, here we go, whenever an analyte molecule comes close to this pore or translocates through that pore, it partially blocks that ionic current. And this leads to these translocation events with a certain blockade depth, so current blockade depth and a certain event duration. And there may be some more fluctuations in these events. All of these observations are protein characteristics which make them interesting for studying proteins. Now there's one big challenge, and this is that in the most simple translocation experiments, your observation time of one single protein is very short. I mentioned here about the milliseconds. That would be true for protein pores. For solid state nanopores, it's even shorter, more in the microsecond range. So one challenge I said was how to extend that observation. No, okay. I don't see the right-hand side of my screen, but fine. So I will present to you in this talk several examples where people found ways to extend this observation time and to much longer than seconds, and that is particularly useful for studying protein dynamics. But first, one more slide on nanopores. So nanopores can roughly be categorized into solid state nanopores and biological nanopores. On the solid state side, we have different types. You can use nanopipettes that are really pulled with the pipette puller until they feature just a very tiny nanometer sized hole. Nanopores can also come on chips, in the most common case with the silicon nitride membrane, and oftentimes drilled with a transmission electron microscope, actually, where you can really drill a hole into this membrane. And then there's no end towards all kinds of more sophisticated nanofabricated pores, including 2D materials, as where the membrane really consists of an atomic or a single molecular layer. Like here, I think it was from a publication on MOS2. On the biological side, we have natural protein pores that incorporate into a lipid membrane. These can, of course, be engineered by mutations to actually tune their sensing capabilities. And in addition, people also engineer by now nanopores using DNA origami, for example, also using peptides. Then, to detect actual nanopore signals, we need a setup, and roughly all, the basic case is that they consist of a flow cell with 2 buffer reservoirs, where in between here you would sandwich your nanopore, be it a biological nanopore incorporated into a lipid bilayer as an insulating layer, or be it a solid state nanopore. In both cases, there are 2 electrodes on either side of this membrane, and amplifier and the digitizer are used to then read out these currents and process them on a PC. There are also by now much smaller implementations of such amplifiers, and Oxford Nanopore also developed other small handheld devices for nanopore experiments. With this, I come now to a few applications of nanopore technology, specifically to protein science and to detect protein functional mechanisms. And let's start with this famous experiment out of Gundlach's lab, where they can really sense how a helicase, a single helicase, DNA processing protein reels in a DNA single strand through such a nanopore. This DNA gets anchored in this nanopore because of its negative charge, so there's a pulling force downwards in the slide, and the helicase reels in now this DNA, which allowed Gundlach and co-workers to really resolve the half base pair steps of this helicase. And of course, this was a big breakthrough, and enabled also later on DNA sequencing, which is now commercialized by Oxford Nanopore Technologies. And actually this year, there was another breakthrough, I think it's fair to call it that way. Just recently, 2 labs managed to sense peptide sequences for the first time in this using or building upon this approach. So basically what they did is they used a similar approach with a DNA processing motor protein, and hooked up this peptide sequence here to the DNA strand, and now the DNA processing motor still processes that DNA piece, but at some point it reels in also the peptide, and this peptide sequence can be read. And in this second paper by Brinkerhoff et al., they even managed to create a situation where repeatedly the sequence can be read over and over again, which of course increases a lot the peptide sequencing accuracy. Another example which has nothing to do now with any kind of sequencing, is from the Giovanni Maglia lab, where they managed to trap a DHFR protein, the hydrofolate reductase, in a protein nanopore really inside, and they can actually detect the functional cycle of this enzyme, how it processes folate, and they can really detect actually up to 5 different states, and how they process, like how they are timed, and all of that. Another example is the one of transient binding. I mean, all of that is detected in a label-free way. Here, Thakur et al. Added a Barnase binding moiety to a protein nanopore sensor, and they can now detect the transient binding of this bar star domain to Barnase. And as you can see, in a very nice way, gives this super nice 2-state signatures. I find also this a very nice example. Then in the end of my talk, I dare to plug in some slides on our newest paper, which is on the nanopore electroosmotic trap that I did as a postdoc in Kees Dekker's lab. How does this work? So here a DNA origami ball is used to induce electroosmotic flows to trap a protein in the most sensitive region of a solid state nanopore. This gives us these nice 3-step blockades. We can then hold such a protein for very long time, so for seconds to minutes actually. We did this for several other proteins. So you see here always some current snippets with these 3 levels then, sometimes more levels even if there's some protein dynamics going on. And when we plot the main blockades of the protein itself versus molecular weight, we find this linear behavior, which also lets us, of course, distinguish these proteins. And the asymmetry of this ClipX protein, which is disc-like, led to a second peak for the 2 different orientations. With this technique, we could also already show different or distinguish different conformations of one and the same protein in a nucleotide-dependent way. So this is HSP90, which occurs naturally in a conformational ensemble in an open V-shaped way, but also in a more compact state, in particular if it is bound to AMP-PMP, whereas the structural compaction is lost towards ATP, ADP, and in the APO case, it's really a very floppy homodimer, by the way. And with these NeoTrap recordings, we managed to detect the different events, and taking a histogram of many such events, we see a very clear shift from this compact state towards much more heterogeneous currents that we read. So this is, as far as we know, the first labor-free detection in solution of these conformational dynamics. Okay, with this I come to my summary. I try to briefly introduce to you the very basics of nanopore technology. Protein-dependent signals can be detected in a labor-free way. There are many diverse types of nanopores. It's really difficult to squeeze this in such a short talk. I try to briefly highlight a few of my favorite applications of nanopore technology. And I ended up showing you our newest work, which is on the NeoTrap. And with this, I come to my acknowledgements. For the work I showed here, was Kees Stekker's lab and Hendrik Dietz and Pierre Stömmer were our collaborators. So these were the most important people for the work I showed here. This is my newly minted small, or the group with whom I work now. And I'm happy to take your questions at the very end of this webinar. Thank you.
Excellent. Thank you very much, Sonia. And our final speaker today is Dr. Peter Hydesen. Peter received his BSc and MSc degrees in biochemistry from the University of Iceland, where he studied the link between enzyme kinetics and flexibility. For his PhD studies, he joined the structural biology and NMR laboratory at the University of Copenhagen, where he used NMR spectroscopy and single molecule optical tweezers to study the protein folding problem. Peter then got involved in a collaborative project between Copenhagen and Cambridge universities, looking at protein misfolding and chaperone action. Subsequently, he studied intrinsically disordered protein complexes with single molecule spectroscopy in Ben Schuller's lab at the University of Zurich. In 2019, Peter joined the Department of Biochemistry at the University of Iceland as associate professor, where his research focuses on using single molecule techniques to study the interactions of transcription factors and chromatin. And today, Peter will tell us about disordered protein interactions studied with single molecule thread.
Thank you, Dominika. Yes. You can hear me and see me, I hope. And first of all, thank you so much for the invitation, and to the Biochemical Society for including me in this webinar. So let me just see if I can get a pointer here. Yeah, so what I want to tell you about today is how we can use single molecule spectroscopy to study the interactions between disordered proteins. And so disordered proteins are quite common actually, and especially in the human proteome, we have a sort of a continuum of protein structures. So I took this picture from a recent review from Julie Foreman-Kay group, and so most proteins, actually 58%, contain both folded domains and intrinsically disordered regions. And sort of the proteins that we have been very interested in are DNA and RNA binding proteins, and they tend to cluster towards the right side of this picture, so they tend to be more disordered than the general population of proteins. And because it is really a major challenge to study intrinsically disordered proteins using classical ensemble methods, we really need an alternative approach. And so single molecule spectroscopy is a really powerful technique to study such proteins. And I just want to tell you a little bit what that's about. So here we have a protein that we've labeled with a donor and an acceptor fluorophore. And so we can excite the donor fluorophore, and then either of two things can happen. So either the donor can emit a fluorescence photon, or it could transfer its energy to the nearby acceptor, and then the acceptor can emit a fluorescence photon of a different wavelength. And really the strength of this method comes from the fact that this process, the efficiency of energy transfer, is very steeply dependent on the distance between the donor and the acceptor. And so if they're far apart, it's like in the unfolded state, so this protein is in equilibrium between an unfolded and a folded state. And so in the unfolded state, the distance is quite large, and then the energy transfer is quite inefficient. And in the folded state, they come closer together, and so then we have a high efficiency of energy transfer. And so this is really on a very useful molecular scale, usually in the order of about 1 to 10 nanometers. And using the transfer efficiency, we can really calculate the distance between the two dyes. And so this really acts as a molecular ruler. And all we have to do is to measure the transfer efficiency is to count the number of acceptor photons and the number of donor photons. And how do we do this in practice? Well, we actually need a confocal microscope, such as the Microtime 200 that we use from PicoQuant. And combined with the fact that we can make a very small confocal volume, so we have here an objective, we have a cover slide, and our sample is here that has fluorescently labeled proteins. So when we have a very small confocal volume and we combine it with the fact that we use very, very low concentrations of proteins, we ensure that at any given time there's only one protein in the confocal volume. And so once in a while, a protein will diffuse through the confocal volume and get excited and emit the fluorescence photon multiple times before it exits again. And so if we just look at the photon counts over time, we'll see that besides sort of the fluorescent background, we'll get these bursts of fluorescence photons that you can see here. And each one of these bursts is a single protein molecule that's diffusing through the focus. And so what we can do is we can take every one of these single molecule bursts and calculate a transfer efficiency. And usually we will put this in a sort of a histogram. And immediately from this, you can see that we can separate at equilibrium the signal from the unfolded and the folded molecules, and then we can analyze them separately. And if we're lucky enough to have pulsed excitation, we can also get information on timing statistics of the photons, and then we have access to lifetimes, and we can extract a lot more information. And so we used these methods. I did this in Zurich when I was a postdoc with Ben Schuler. We worked a lot on these with these methods on intrinsically disordered proteins, and this is sort of what I'm continuing on here in Iceland. And there we studied very heavily disordered proteins. So histone H1 has a small globular domain that you can see here, and it's flanked by these long intrinsically disordered regions, and it's very positively charged. And we were studying the interaction to prothymosin alpha, which is its chaperone. And this is a very negatively charged and very disordered protein. And so I just want to boil down really what the story was that we published a few years ago, where we used a combination of single molecule FRET, NMR, and simulations to show really that the complex between these two proteins is really, really disordered. So they don't have any fixed really structure in the complex, but they bind really tightly together in the picomolar range. And so this prompted us to ask the question, what is the functional advantage of such a complex, and why does it really need to be so disordered? And in order to answer that, we sort of need to take a look at what is the function of these proteins. Histone H1 binds nucleosomes, and so you can see here a structure of the nucleosome, and you can see that the globular domain sits here on the dyad. And we don't know, I mean, in the structure, you don't resolve the conformations of these vast disordered regions. But when histone H1 binds to the nucleosome, it sort of collapses these linkers that connect adjacent nucleosomes. And this leads to a global collapse of the chromatin fiber, so you get this condensed chromatin. And so really what we wanted to start with doing was to understand what are the conformations of the disordered regions on the nucleosome, and to do that we used single molecule FRET. And we really mapped the histone H1 nucleosome complex very thoroughly using FRET. And we did that by placing a donor dye in any one of 6 positions along the sequence of histone H1, really probing the entire polypeptide. And we placed an acceptor dye in any one of 8 positions along the nucleosome structure from the linkers to the core, and then we measured FRET between all of these labeling pairs. And what we end up with is this sort of a distance grid, so we can convert the transfer efficiencies to distances. And just to summarize what we see from this complex picture actually is that qualitatively we can already say from this that histone H1, the disordered regions, don't really have a fixed structure, but rather they sample a very broad distribution of distances in a dynamic manner. So we turned to simulations, and so we used coarse-grained simulations to try to simulate the complex and see really to get a sort of a molecular picture of what it looks like. And I don't really have time to go into the details of this model. I mean, we essentially treat the entire system as a string of beads to simplify things. So it's a very coarse-grained model, but a very simple model that really is dominated by charges, is able to reproduce the FRET efficiencies from the experiments that you see in blue, and the simulation that is in gray really reproduces the pattern and the absolute FRET efficiencies of all these labeling positions that you see here on the x-axis. And so what does the simulation look like? It looks a little bit like this. This is about a microsecond that is condensed into about a minute. And what we can see is that histone H1 is really disordered and dynamic on the nucleosome. We see the globular domain that sits here on the dyad, as it should. And the disordered regions really sample a very broad distribution of distances. And so the other thing that came out of this when we did our binding studies is that the residence time of H1 on the nucleosome was in the order of hours. And this was really incompatible with efficient regulation in vivo. And we already know in vivo that the residence time is in the order of minutes. And so we suspected that the chaperone that I told you about, prothymosin alpha, may have something to do with reducing this residence time. And so we turned to experiments on the surface. And so here we immobilize nucleosomes on the surface, and they are fluorescently labeled with the donor and acceptor. And we have H1, which is here in blue, freely diffusing in the solution. And so when you bind H1 to the nucleosome, it collapses these linkers, bringing the dyes close together, and then you have high FRET. And then when H1 is unbound, the sort of linkers come apart, and then you have lower FRET. And so if you look at the fluorescence over time, you see that the acceptor fluorescence is higher most of the time, indicating that it's bound. And then you have these occasional dissociation events. And then we just add prothymosin alpha to the mix and see what happens to the kinetics. And when we do that, and even just at low micromolar amounts, we see that the frequency of the events, these dissociation events, increases. And it increases even further when we go up to about 100 micromolar, all the way to the point where we see mostly the unbound state. So obviously prothymosin is doing something here. And when we plot these rates as a function of prothymosin concentration, we can see that the data actually is only consistent with a model that involves a ternary complex, meaning that prothymosin alpha invades and forms a ternary complex to really enhance the dissociation of H1 from the nucleosome. And so we can also add prothymosin to a similar coarse-grained simulation and show that it indeed does bind histone H1 on the nucleosome, and it enhances the dissociation of it from the nucleosome, which is very consistent with the kinetics that we measure in our single molecule FRET experiments. And so disorder really enables an acceleration of H1 dissociation from the nucleosome by almost 2 orders of magnitude. So that's quite a big effect, which can explain in part why the residence times in vivo could be so short, because you have chaperones and you have all kinds of other things that actually take part. And so I just want to leave you with key points. I mean, that was an example of how we can use single molecule FRET to tackle very complex problems. Single molecule spectroscopy is a really powerful technique to study distances and dynamics of biomolecules. It enables access actually to molecular timescales that span over 15 orders of magnitude. So the experimental toolbox is very, very large. We have correlation spectroscopy and the kinetics and microfluidics and all kinds of things that we can do to study different molecular timescales. I've shown you that it's actually ideally suited to study the interactions of disordered proteins with chromatin, and this is what we're doing here in Iceland, taking these concepts a little bit further. And I showed you specifically that single molecule FRET really revealed that the release of linker histone H1 is driven by a polyelectrolyte competition with a disordered chaperone. And with that, I'd just like to acknowledge, I mean, this was all of this work was done with Ben Schuler in Zurich. David de Mercadante and Robert Best did some of the simulations, and all of these people came into this in one way or another in very challenging work. And these are the funding agencies. And thank you for your attention.
Excellent. Thanks very much, Peter. So now I think we can welcome questions for all the speakers. And so if you have a question and you haven't done so already, please type it in the question box as shown in the image on the screen. And in fact, let me just unhook them. We have 2 questions already from one very active member of the audience. So George, I think we're going to go with you first. So the question is from Deb, and Deb asks, in the single molecule replication reaction, how far the protein molecules should stay with respect to each other on the DNA strand in order to capture them as individual molecules in the TERF microscope? So okay, I think it's question. A spatial resolution question. Yeah.
Yeah. So the molecule is about 50,000 base pairs, the lambda DNA, and I think each pixel in the microscope is about 500 base pairs. So you can only really distinguish proteins that are over 500 base pairs and more like over 5,000 base pairs apart. If that makes sense.
Yeah. Okay. So now let's move to Sonia. So that's also a question from Deb. Thanks very much, Deb. So the question, can NeoTrap be used to study sequential multiple protein-protein interactions by adjusting the diameter of the nanopore?
Well, here I guess I would have, first of all, thanks for the question. I would have a return question. I'm not sure if that works. But, what is meant with sequential in situ, I guess, is the question. So for the size of the nanopore can be tuned within, well, at some point we would have to make a bigger DNA origami sphere than we have right now. Right now the sphere is 35 nanometers in diameter, and we worked with, what we did already so far was we worked with nanopore diameters of up to 30 nanometers. And there's always a coating, so this ends up being a 20 nanometer coated pore. The interactions could be sequentially detected if they lead to distinguishable signals, and this is something that's very difficult to predict beforehand. So that's actually quite often in these nanopore experiments. It's different from, for example, FRET spectroscopy, where you know what kind of distance dependence you can expect and how this will be reflected in your data. That is a bit is often more complicated in the nanopore data. So what people usually do is they build up their experiments in a systematic way, just adding first one analyte, then a second analyte, a third analyte, and to see which interactions occur then that were not visible beforehand. And like this, you can infer where the signals come from. So I cannot give such a generalized answer. I think there can be situations where this is possible. Yeah. That I think is as good as I can answer.
Excellent, thank you very much. Can I just ask Sangamesh, who asked a question, to perhaps indicate who this question is addressed to, so that I can ask it later. And in the meantime, I'll probably follow up with a question to Peter. So I actually want to ask, because that's very intriguing, and I guess this is the first fuzzy complex that I can truly believe in. But I have a question with the nucleosomes experiments. Have you tried to do these, and I know they're very difficult, in the context of dinucleosomes, and what would happen then with the H1 binding? And
that's a very good question, and actually we sort of flirted with these experiments, but we took it a little bit more, further than a dinucleosome. We actually went straight for a 12-mer. So we went from a mononucleosome, and then we thought, well, let's just go all the way and just go big, and we went for a 12-mer. But as you said, I mean, this is really technically demanding, and especially with H1, because it doesn't, so it doesn't really have a specific binding site, so it'll just bind to all of these regions, and on the single molecule level, this can be quite difficult to decipher. But what I can say is that we did manage to do some tricks to at least show, and this is not as part of the paper or anything that's coming out now soon, but we did show that the overall dimensions of the disordered regions is the same in a 12-mer, even when it's compacted by when you have many unlabeled also H1s in there. But the disordered regions essentially have the same dimensions, which is in a way surprising, actually, because you have a completely different environment. But I think the reason is that even in the mononucleosome, you have enough DNA there to really saturate the contacts of all these sort of electrostatics that are going on. And so adding more DNA to that doesn't really make a difference to the overall dimensions. And so this becomes kind of like an electron cloud in a sense. But yeah, we did flirt with that, but we never got far, unfortunately. But this is entirely what I'm trying to do more of here. So moving into bigger systems and looking at sort of di, tri, tetramers, tromers, and beyond.
Okay, excellent. I have lots of more questions to all of you, actually, but I'm going to crack on. So Sangamesh actually came back saying that was the question for you, Peter. And so the question is, how about tracking changes in orientations along with the distance?
Tracking changes in orientation. So you mean then, so one of the things that we did, which we can do, well, we did with the simulations, which is to look at how the globular domain sits on the nucleosome. Maybe that's what you're talking about. I hope that's what it is, because essentially in our experiments, you're probably right about this, that we can't discern whether it's turned sort of this way or 180 degrees on the nucleosome, on the dyad. But we did see indications in our histograms that we saw this as well, because we would see in some labeling positions evidence for this. And when we did the simulation, we essentially used the crystal structure of the nucleosome, but then what Davide did was to do 180 degree of the globular domain, and then he could actually reproduce some of those additional populations that popped up. And so we couldn't really quantify how much was in each orientation, but I actually think that generally speaking, they were almost equal. But we didn't really go more into this. But sure, there are, of course, other labeling strategies that you could go through to really pinpoint what these orientations are. Yeah, that's a good idea.
Okay, so can I ask a question now from myself to Sonia, actually? So in the protein nanopore experiment, the protein sequencing sort of, what is the length of the readout? How long can they go for? Because I haven't seen these papers.
Yeah. Indeed, that's limited, right? Because they still rely on processing of the DNA.
The DNA.
If I remember correctly, it was up to, I saw a sequence up to 15 amino acids, which actually sounds quite long, but definitely will be possible. So this will be will remain a limitation for that experiment, but what is already done also in today's peptide sequencing is that people rely on the overlapping sequences, right? That you then can add together the bits and pieces and resolve the entire sequence of a protein. I guess this is what people now have as a next goal, to really go far beyond this limitation of these very first experiments that are now published.
Excellent. Okay, and last question for George from me. So how do you think, and I know I must have asked you this question before, but how do you think RTL and FuncJ actually drive the eviction of MCMs?
Yeah, it's a good question. I think, I think a model I quite like is maybe these helicases acting on the other side of the replication fork could help sort of pull the DNA actually maybe through one of the interfaces between the MCM hexamers, like between the subunits in a single hexamer, and that could help remove it from the DNA. And I think that wouldn't happen with CMG, because we sort of know that CMG isn't a very strong helicase, whereas these helicases might be stronger and would allow that. I mean, the other thing is maybe if they're sort of, if you imagine all of my diagrams are showing just like naked DNAs, and if a double hexamer sliding on a naked DNA, that's not really happening. There's nucleosomes and other DNA binding proteins present. So maybe kind of helping push against something that's already bound to the DNA could cause them to collapse. But I guess what we do know is you do need something strong and active needs to happen, because we know that these double hexamer complexes are quite stable. You can add half molar salt to them on DNA and they will remain on DNA. So yeah, something active.
Yeah, something along those lines. Okay. Yeah. All right, one last question, quick question to Peter. Why does SM-Fred is better suited for disordered proteins with chromatin? Why chromatin?
Yeah, that's that's also a fuzzy answer actually, because why I say it's ideally suited is essentially because size-wise, I mean, there aren't so many methods that actually are great for studying disorder. I mean, disordered proteins are really, as I said in the beginning, like a major challenge. I mean, it's really challenging to understand them. And so one very powerful method is NMR that gives you really high resolution information, but then the size of the system becomes difficult. You know, so there's a group, of course, that does, so Hugo van Inkel does nucleosome work and on the histones, and he's done some beautiful work. But when you want to increase the size a little bit further, then NMR becomes quite difficult. And I think we have a lot of potential using the fluorescence strategies that we have to really look at big targets, you know, look at really nucleosome arrays, and to use sort of the single molecule fluorescence techniques that we have to build really accurate models actually of what's happening. And so I think in terms of chromatin, I'm not saying single molecule FRET necessarily, but single molecule techniques in general would be very, very, very powerful to study disordered interactions with chromatin in that respect, I think. Yeah.
Excellent. Okay, well, thank you very much. And I think, given that we just want to run out of time, I think I would like to thank all the speakers one more time for a great scientific session. And of course, I would like to thank Pico Quant for sponsoring this webinar. I also want to thank the Biochemical Society events team for the work behind the scenes. And my special thanks go to Sandra, who has made this event happen and made sure that everything ran smoothly for everyone. And of course, finally, I would like to thank our audience for joining us today and being engaged and asking questions. You can, of course, continue the conversation online by following the Biochemical Society and Portland Press on Twitter. Also, you can find more information about careers, including career profiles and job-seeking advice on our website. You can also find some day-in-the-life career profiles on the Biochemist webpage. Of course, we welcome suggestions for future topics and speakers to feature in our Biochemistry Focus webinar series. So if you have an idea for a webinar in 2022, we invite you to submit a proposal for an upcoming webinar. Now, if you have missed any of the 40+ webinars that we've run as part of the series or would like to watch them again, please visit our website or YouTube channel. And the recording from today's webinar will also be available to watch, hopefully within the next couple of weeks. So you can find more information about the webinars, how to propose your webinar, and watch previous recording on biochemistry.org/webinars. So please join us for the next webinar in the series entitled Environmental Sustainability in Biomedical Laboratories on Wednesday, 24th of November at 2 p.m. GMT. During this session, chaired by Dr. Ben Foster at the University of Oxford, we will hear an introduction to the LEAF platform, followed by a talk from the sustainability team in Oxford about setting up local infrastructure for sustainable labs, and the lab manager directly involved in carrying out sustainable labs initiatives. So finally, I'd like to highlight that in these challenging times, it's more important than ever to stay connected and engage with your fellow molecular bioscientists. It's an extraordinary time for us, but also it's an exciting time to join the Biochemical Society and our community to stay connected and take advantage of key benefits, which include discounted registration fees for society courses and meetings, exclusive access to a wide range of grants and bursaries, personal online access to 2 of their journal, and many, many more. And so please visit the Biochemical Society website to find out more about it. And finally, thank you very much from me, and goodbye.